Thank you for having me. It's wonderful to be here. So, as Sonia said, I'm going to talk about glass transition in epithelial tissues. And my name is Sumantra, and I come from IISc, which is pictured right over here. And they are the one of the main funders of this work, as are all of these funding agencies that are listed over here. So I'm very grateful for them to support our research. So this particular work has been part of an ongoing and wonderful collaboration between a colleague of mine, Medhavi Vishwakarma, who is down the street in the bioengineering department at ISC. And in today's talk, whatever experiments I'm going to show, it is done by Medhavi and her incredible student, Sindhu, who is in the postdoc market. And the theory part has been done by my group, mainly by these three people, Shomadev, Panindra, and Lakshman. Shomadev and Panindra are PhD students, and Lakshman is an undergrad in physics. And this work has recently been published. In one contains mostly the experimental aspects, and another contains mostly the theoretical aspects. So if you are interested, these papers are listed over here. And so let me put the laser pointer. Okay, cool. And but if you want to know more about my group and what we do, you can visit my website and our group's website over there. So let's get started with the topic of the talk, that is glass transition. And in particular, glass transition has been studied for the last 50 years or so. And so we have come to a good consensus about what we mean by glass transition, in particular in the context of passive systems. And what are the minimum requirements of the glass transition is a discontinuous freezing transition, such as the one that is shown over here. At some temperature Tm, a liquid discontinuously goes into a crystalline phase if you allow it to cool it sufficiently slowly. And this rate is called the nucleation rate. However, the trick is that if you cool the liquid faster than this rate, it will go into what is called a supercooled liquid phase. And then that supercooled liquid phase can avoid this crystalline trap and eventually get into this glassy phase. So what is interesting about the glass transition is that people have been debating about the true nature of this transition, whether this is a phase transition or not. But for now, what is important to know is that when glass transition happens at the temperature, that actually depends on how fast you cool the system. The faster you cool, the sooner you get the glass transition. And this is quite important, a point to remember. I mean, the same thing you can do for colloidal system, like hard sphere colloidal systems, where there is no concept of temperature, but you have what is called packing fraction. And you can use the packing fraction in the same manner as temperature for molecular glasses to drive glass transition in colloidal systems. But irrespective of the system that you choose, there are certain hallmarks of glass transition. And one of the is diverging relaxation time, which perhaps many people know that as you approach glass transition, the viscosity increases by almost 14 orders of magnitude. But there are other hallmarks also, such as dynamic heterogeneity, in which you have coexisting liquid-like and solid-like state at the same density, as shown over here. So for example, some regions are flowing, whereas some regions are not really flowing. But perhaps the most important, or rather like the smoking gun evidence of glass transition, is what I would call aging, which is shown over here. And what does aging mean is that you basically, any property that you measure, such as say a correlation function, it depends on when you start measuring. The time origin of the measurement does play a role. And in particular, if you sort of keep on increasing the origin, your correlation functions start to develop this shoulder, which basically tells you your system kind of gets caged as it matures into this glassy phase. And these are all important features of glassy behavior, and many of them have been found in many epithelial tissues experiments. So one of the classic demonstration of this was done by Tommy Angelini from Jeff Redberg's group back in 2011, who showed that there are dynamic heterogeneity in this system that depends non-trivially on the number density of the cells in the tissue. More recently, what people have shown that it actually, there are features of aging. So for example, this is an experiment again from Fredberg group, but done by Jim Park, who showed that at constant density, if you allow the epithelial tissues to mature, if you measure something like, let's say, 4-point correlation function, 4-point susceptibility, it actually shows can actually show a history dependence and aging. But there are many other experiments, with some of which are listed over here. But the key idea that these experiments sort of tell you is that cell division-induced crowding does drive glassy dynamics in most cases, and that there is a threshold density above which the epithelia is glassy, below this density, it is fluid. And clearly, like, this requires some kind of a theoretical understanding. So people have been trying to understand it for a while. And the one of the most successful theories, unfortunately, or rather fortunately, has been done in the absence of any density dependence. So Max B and colleague, what they showed was some remarkable result that if you take an epithelia in a model called the Vertex model, which I'll define in a slide or two, uh, what you can show is that the transition from a fluid-like state to a solid-like state is driven actually by the shape of the cells in the tissue instead of anything else, because these are confluent, so there is no concept of packing fraction, and because these are active system, there is no concept of temperature either. So the shape, there is this particular thing called shape parameter. It can drive fluid to solid transition. Max later showed in a different context that using a different model called the self-propelled Broglie model, which can be, but it can be shown for the Vertex model also, that if you add cell motility on top of the cell shape, still at constant density, you can create this two-dimensional phase space in which in some part you get fluid, in some part you get solid, but you can actually get glass transition if you pick some points carefully. So, but the key idea here is that it is density independent. So they are all being done at constant density. But as I have shown you that in most of these experiments, the experiments actually, you have proliferating tissue, which means the cell density is constantly changing. And the question then is, does this result survive if you allow the density to change? So what I'm showing you is a simulation of the Vertex model in the presence of cell proliferation. And as you can clearly see that as the cell density progresses, the system eventually becomes very, vortex-like and flowing. So what that basically tells you that cell division essentially leads to fluidization in any of this system. And this doesn't really matter like which point you choose in the parameter space that I have just shown you, it will always lead to fluidization. There is a very interesting reason, but I'll come back to it later, why this happens. But the interesting thing to contrast is this experiment by Sindhu, in which you can see that initially the cells are big and they're very fluid-like, but as the tissue matures through proliferation, you start behaving very much solid-like. The motion completely gets arrested. And what is even more interesting is that if you look at the Vertex model, the cells sort of become highly anisotropic as the density increases, whereas in Sindhu's experiment, the cells become more and more isotropic. So clearly, there is some issue with the canonical Vertex model that is preventing us from reproducing what we are observing in the experiment. And that was the first thing that we wanted to understand, like how do we get tissue solidification through cell division from a modeling perspective? As I've been telling you, Vertex model is the model of our choice. And what Vertex model has is this energy function, which has 4 parameters. There is this bulk modulus K, the preferred area of the cells, A naught, the perimeter modulus gamma, and the preferred perimeter P naught. And AI and PI are the area and perimeter of the ith cell. And the sum of all the cells would give you the total energy of the system. And the way you would simulate it is by going along the gradient of this energy functioning. But the important thing to note is that there are certain combination of these parameters through which you can write down contractility, which is given over here, and line tension, which is given over here. They are the ones that are ultimately deciding the fate of the Vertex model tissue. And what is important is that both of them arise from the organization of actin. So this is something that you can relate to experiments. In particular, Paradifer and Staple, who originally worked a lot with the Vertex model, they came up with this phase diagram in terms of contractility and line tension, in some part of which you get fluid-like behavior, in another part of which you get solid-like behavior. But what is interesting is that now you can actually relate this to the actin morphologies that are present inside the cell. So for example, the contractility can be related to the stress fibers. The more stress fibers you have, the higher is the contractility, whereas the more junctional or cortical actin you have, the more is your line tension. And usually in the canonical vertex model, you pick one or the other point. And what that means that you choose certain relative abundance of the stress fibers and the cortical actin that ultimately decides the fate of your tissue. But if you actually now look at the experiment done by Sindhu, it's kind of very interesting. So what I'm showing you here is a snapshot in which the actin cytoskeleton has been fluorescently imaged. And if you look closely, what you find is that at low density, low cellular density, there is a overabundance of the stress fibers, whereas junctional actin is very little. And at high density, there is hardly any stress fibers, whereas the junctional actin is quite rich. And what that tells you is that the density is sort of driving some kind of a chemical change in the actin cytoskeleton, which is driving the complete architectural reorganization in the actin cytoskeleton itself. And there is this kind of a mechanochemical feedback, which is present in the tissue as the tissue matures through proliferation. And ours is not the only work. There have been some other work, recent works, that have also shown the same thing. But the upshot of all of this is that if I go back to this picture, it just tells you that the tissue that we are seeing in the experiment no longer resides on a single point in this parameter space, but rather actually follows a trajectory as you are increasing the density. In particular, in Sindhu's case, at low density, you will be roughly over here where you have more stress fibers. And at high density, you will go roughly over here where you have more junctional actin. And this is something that we need to capture into the model. And we can capture this by considering the load-dependent or density-dependent binding of myosin in active actin. So what myosin does, it has load-dependent unbinding from actin. In particular, the unbinding rate actually reduces as you compress the system. And the way you can capture this is by defining this areal strain, A minus A hat by A hat, where A hat is inversely proportional to the binding affinity of myosin to actin and is itself a reference area. And when you do all the calculation, what you can find is that the fraction of the junctional actin has this nonlinear dependence on the cell area and cell density. 1 by A is the density. And in our sort of two-state model, 1 minus P bound then basically then gives you stress fibers. So, and the feedback strength of this mechanochemical feedback is then given by this A hat. So this is how a typical P bound looks like as a function of cell density. So as you can see, the fraction of the junctional actin increases with density, and its conjugate, the stress fiber, should decrease with density. So that means that we can sort of capture the behavior that we are seeing in the experiment, provided that we make the following constitutive relationship. And the way you make this constitutive relation is that by noting that line tension is equal to minus gamma times P naught, and we expect line tension to be proportional to the concentration of the junctional actin, which means P naught is proportional to 1 minus P bound, and the contractility is proportional to inverse of A naught, and that it essentially tells you A naught is proportional to P bound. So when you put this thing together in this picture, you go from low density to high density in the fashion that we have seen in the experiment. So this is quite encouraging. But remember that glass transition or tissue solidification, the way I've shown you, is a dynamical behavior. And to consider the dynamics of the tissues, we also have to consider one more thing, and that is motility. Okay. The motility also has density dependence. And in particular, in this paper by Jacob Nottbaum's group, what they had shown that stress fiber not only controls the contractility, it also controls the motility of the tissues. And in particular, the more stress fiber you have, the more motile the tissue is. And what that means is we can actually write down this constitutive relation in which the motility is proportional to the fraction of the stress fibers, which is 1 minus P bound. So as a result, with respect to density, the average motility actually goes down non-linearly with density. So what that means, if I go back to this picture by Max B, again, we are no longer picking one point in this phase space, but rather we are following a trajectory in this phase space as we are changing the density. And our hypothesis is that essentially by going along in this three-dimensional phase space, which include the V naught, contractility, and the tension, that we are going towards a solid state as we're increasing the density. So to put this idea into test, we put all of these constitutive relation into a vertex model, and this is how it looks like. So as you can see, it is clearly becoming more and more solid and isotropic as the density increases and the proliferation increases, which basically gave us the confidence that we are on the right track. And so what is even more interesting that people who work on this field, there is a really important thing called a shape index, which Max B basically figured out. And what what I'm showing you here is the shape index as a function of density. So what you can see clearly is that there is a density at around 1.5 at which the yield stress becomes positive. That's the line at which yield stress is zero. Above this, you get pure fluid. Below this, you get solid and fluid coexistence. And eventually, at high enough density, you get into what is called the pure solid phase. And this is quite interesting because now this is telling that because there is fluid solid coexistence, it can actually lead to glassy behavior. So that is the question now we want to understand, like whether the solidification that we have seen, whether this is glass transition or not. Okay. And to understand whether this is a glass transition or not, we want to go back to this picture that's Sullinger and De Benedetti painted back in 2001. But we want to sort of adapt it to the epithelial tissue case, in which the claim is that the transition is driven by cell density, right? So if this is true, then we should have a picture like this, in which as we increase the density, in say quasi-static manner, then it should go from the liquid to fluid-like to the solid-like state in a discontinuous transition. But if we increase density through cell division at a finite rate, then it should go into a glassy phase, and then at a higher division rate, it will get into the glassy phase faster. So that is the claim. So let's see what we get from our simulations. So this is our simulation result. So let's focus on the maroon curve right over here. So the maroon curve basically shows you the quasi-static limit of the model, in which we fix the density and allow the system to reach its minimum, energy minimum. And what you see is that up to some density, the pressure fluctuation is quite high, which signifies a fluid state. But at some particular density, there is a discontinuous change in the fluctuation, which then signifies transition to this kind of a solid-like phase. But interestingly, if we now do the same transition in the density with finite division rate, you find that it kind of avoids this discontinuous change. And in particular, the higher the division rate, the faster it kind of gets into this solid-like phase. Okay, so which is shown by this arrow right over here. So what you can do is you can actually probe this hypothesis that it is like a glass transition by using the usual hallmarks such as mean square displacement as a function of the division rate. And as you can see, at higher division rate, you kind of get into this plateau region, which signifies caging events much faster than at lower division rate. So which seems to make this analogy even stronger. But as I said, like the smoking gun evidence of the glass transition is aging. And the way you can prove this in the case of tissue is by defining something called the self-overlap function. So what the self-overlap function does, it basically checks whether the cells in the tissues are caged or not. And the way you do it is by tracking the location of the cell centers over time. And if this is within some cutoff radius, you say that the index function is 1. And if it is above that cutoff distance, you say it's 0. And then the self-overlap function is just an average of the index function of all the cells. And what I'm plotting here is the self-overlap function with some particular old t origin as a function of different division rate. And as you can see that the overlap is significantly higher at higher division rate than at the low division rate, which basically tells you that the systems are much more caged, much more glassy-like at higher division rate, which again corroborates the story that I've been telling you. But here comes the final sort of the proof that this is indeed a glass transition in which what we do is we take the R equal to 100 overlap function, and then we measure it as a function of different time origin. And as you can see, the longer we wait, the flatter it gets, which basically convinced us that this is indeed a glass transition that we are observing. Okay. So that's cool.
Just to let you know, about five minutes or so.
Okay, excellent. Okay, so but the thing is that in the experiment also we see the same thing. You wait longer, the overlap function actually becomes flatter. Okay. So let's come back to the last thing, which is the dynamical heterogeneity. And what Angelini showed that the size of the dynamical heterogeneity has this really interesting behavior in which, as a function of density, if you measure the size in terms of number of cells, it kind of increases and then flattens. But if you measure in terms of the area, it increases and then decreases. We actually find the same behavior from our simulation in which as a number of cells, it increases and then saturates. But if you measure in terms of the area, it increases and then decreases. And what is interesting is that the area at which it peaks is also the density at which the yield stress becomes non-zero. And that is interesting because it signifies that there is a relationship between what you are sort of predicting from the mechanical aspect is something that you're also getting from the dynamical aspect. And that is something interesting because what we eventually then figured out is that the dynamical heterogeneity is correlated with density heterogeneity. And this is not true if you look at molecular colloidal glasses where the density is uniform and dynamical heterogeneity sort of arises because of the dynamics. So in this way, the epithelial glass transition is quite different from the type molecular or the passive active glass transition that people have been studying. And this again arises because of the density dependence of the motility, and higher the density, the lower the motility, and that's how you get this heterogeneity. So what that tells us is that if we understand the origin of the density heterogeneity, we should be able to predict the dynamical heterogeneity of the system. And the way we did that is by measuring the modulus of linear elasticity of the tissue using this constitutive relationship. And what we are plotting here is the shear modulus G, the bulk modulus B, and the P-wave modulus, which is the sum of B and G for 2D, okay? And this P-wave modulus is really important because when it becomes negative, such as over here, it basically signifies the onset of contractile instability into the system. What that means is that a tissue that was uniform in density would no longer stay uniform beyond this density, and it will sort of disintegrate into regions, not disintegrate, but reorganize into regions of low density, which is painted in orange, and regions of high density, which is painted in blue. So that then basically explains where does the density heterogeneity arises. It also tells you something really interesting. It tells you that it is not only just a motility, but if there is anything, any other dynamical behavior that sort of couples with the density heterogeneity, then that should also show some kind of a dynamical heterogeneity. That will be another form of dynamical heterogeneity that we can find in this system. And this is something that people have been studying for a while, so which is arc oscillation or arc wave in the context of tissues. And what people have shown is that there is this negative feedback loop involving a protein called arc and the actomyosin network that are present in the cell that basically leads to sustained oscillation in the tissue. And that is involved in collective cell migration and whatnot. But what is important here to note is that as the tissue is maturing, it is also coupling to this contractile instability. And as a result, whenever you have this high density, it undergoes something called compression-driven oscillation death because of the fact that arc is a Hopf oscillator. And whenever you have low density, it actually continues oscillating. And through that, what we predict, what our theory predicts is that there should be some kind of heterogeneity in arc oscillation in the tissue. And when Sindhu looked at her tissues, she did find that this kind of heterogeneity exists. In particular, regions of high density had really long period, which means like their oscillation was damp, whereas regions of low density had very high period, which means the oscillation was persistent. Okay. And so with that, let me sort of conclude. So the key conclusion of this talk is that, well, the class transition is indeed driven by cell density. Cell density is the driver, but crowding alone is not sufficient. You also need something like mechanochemical feedback, which I had shown in the form of load-dependent binding of myosin to actin. And what is important is that the glass transition in tissues is different from conventional glass transition in the sense that there is density heterogeneity, which drives many of the dynamical heterogeneity observed in the system. And hopefully that will shed light on many of the observed behavior in the system. With that, I thank you for your attention, and I'll be happy to take more questions.